Good afternoon. My name is Sean Horrigan, Director of Communications for the Rappaport Foundation. Thank you for joining us for the 2022 Rappaport MGH Research Scholars presentation. This afternoon, we are joined by Drs. Leonora Balaj, who will present on using blood-based tests to diagnose and monitor brain tumors, and Dr. Shabani Mukherjee, who will present on optimizing treatment strategies for people living with infectious diseases and depression. Each of our speakers will present for about 20 minutes, and we will allot the remaining portion for Q&A. Before we begin, let me give you some background on both of our speakers today. Leonora Balaj is an instructor in neurosurgery at Massachusetts General Hospital and Harvard Medical School. She received her BS in biology from the University of Bridgeport in Connecticut and her PhD in medicine from the VU University in Amsterdam, Netherlands. During her graduate studies, she worked in the laboratory of Dr. Bregfeld at Massachusetts General Hospital in the Department of Neurology. Her work focuses on the characterization of brain tumor-derived vesicles as a tool for cell-to-cell communication, as well as a source for biomarkers. Dr. Shabani Mukherjee is the Associate Director of Neuroinfectious Disease Unit at MGH. Her clinical focuses are on neurology infectious diseases with a specific interest in neurological complications and aging in people with HIV. She received her undergraduate degree from Yale University and her MD and PhD in neuroscience from Case Western Reserve University School of Medicine. She completed an internship in internal medicine at MGH and a residency in neurology at Massachusetts General Brigham Harvard Neurology Residency. Her lab focuses on HIV pathogens in the central nervous system. So why don't we begin with you, Dr. Balaj? Thank you. Please take it away.
Wonderful. Thank you, Sean. Let me see if I can share the screen. All right. Can you see the full screen?
Yes.
Oops. This is better?
Yes, great.
Okay, so thank you, Sean, and thank you to the Rappaport Foundation for having me today here. It's an honor to be here and share our work on developing assays for glioma monitoring and detection. So I'll just briefly go over a little bit what liquid biopsy is and why we're all so excited about this, and why we're using liquid biopsy in gliomas, and how that can be helpful to the clinicians and to these patients, and a little bit more into details about which exactly analyte or what part of that biofluid we're using, some of the challenges in getting these things to work. Obviously, they're not as easy as we would hope that they are. And then finally, a little bit about our assays and what they can actually do once they are used in these patients. And then finally, how we're taking this work into the future. So what is liquid biopsy? Liquid biopsy is similar to tissue biopsy, or the same concept, except that in this case, we're actually using a liquid instead of a tumor, instead of a tissue. So in this cartoon here, you can sort of get the idea. If this is a blood vessel and you have into the tissue something happening, in this case, we're dealing with a tumor, but it can be a disease also, or some sort of other form and inflammation. All of these cells are obviously alive, and they're secreting a lot of information into circulation, into their surroundings. And this information ends up in the blood stream at some point. And these things they're called proteins, they're called exosomes, they're called DNA, RNA. There are multitudes of them that end up in the bloodstream. And then once we take a blood sample, we're able to look for them and to try and understand what is a tumor, or what is that disease telling us, and what is going on at that point in time and at that disease. Then we take this blood, obviously, we go to the lab and we'll look into that. A little bit more about, again, in general, liquid biopsy. So as we know, normally, again, the way to finally confirm a diagnosis is using a tissue. But if we move away from liquid, this would be a non-invasive way to look into that disease. And so far, the biofluids that we can use are several. Blood, you can imagine, the most common, but people have used urine, depending obviously on the disease that you're going after. You can use sputum, obviously cerebrospinal fluid for if you're looking to the central nervous system, ascites, teardrops. There are researchers also looking at these vesicles or the information coming directly from the tissue, so you can dissociate that tissue and looking for specifically for information. So when is liquid biopsy useful then? It's many occasions. Obviously, the easiest one to think about is when you don't have enough tissue available, or when not enough tissue is available in a specific sample for whatever reason. As it happens in glioma, a lot of times the tumor is very hard to reach. In some cases, it's even more difficult because actually you don't even want to go into those regions because you'll cause a severe deficit to that patient. So several times it happens that the biopsy is not even taken into certain patients, and the diagnosis is based on imaging or other methods. More importantly, obviously, when you need regular monitoring, so to be able to say, is that disease or that tumor returning, or is it a response to an inflammation to the therapy that's looked into the imaging? And so liquid biopsy can help to answer all of these questions. And finally, I'll just make another point here for regular monitoring. We're in an era right now where a lot of personalized therapies are being developed, which is great. But personalized therapies rely on the fact that we need to know what's happening in the primary tumor. And if we're not able to know, because we cannot go into the tumor tissue every day or every week or every month to perform a biopsy, obviously this is where liquid biopsy would be so helpful, and also open new ways to treat and test new treatment and treat these tumors if we're able to just use a blood sample. So the things that we're looking for into these biofluids are, as I mentioned earlier, they are plentiful, but include cancer cells, DNA, and I'll go more into details on that. So this is one of those analytes that we look often for in biofluids. And this is what you would call a vesicle, and it looks similar to a cell, except that it's a lot smaller. And so you have all sorts of microRNAs in there, mRNAs, different types of RNAs. You have different types of proteins. You have different types of DNA. So this would be what we call the extracellular vesicles, or the exosome. And so you will have the evRNA associated with it that contains certain information. You'll also have RNA and DNA that also is associated with this information here. And in parallel, you may have cell-free DNA, which is coming also from tumor cells, but may not necessarily sit on these vesicles. It may just be floating around freely in biofluids. So we can look at cell-free DNA, we can look at RNA within these vesicles, we can look at RNA and DNA within these vesicles. Less likely that you'll have free-floating RNA, because RNA is a very unstable molecule and it will degrade very easily. And that's why when we're going after RNA for certain diseases or certain questions that we're trying to answer, typically we look into these vesicles that contain the RNA stable within in circulation in all these different biofluids that I mentioned earlier. So how do we use this information? What do we do with it? So specifically when it comes to vesicles, as you may, as you saw from the previous slide, they contain a lot of information. So theoretically, we can really try to answer a lot of questions from these vesicles. And they're secreted while the cell is growing. The cell doesn't need to necessarily be in trouble or be a dying cell. And so we can gather information from a growing cell, from a dying cell also, but also from any other status of the cell that the cell is going through. Some of the analysis that we look for, we can look for example, if this is that lipid bilayer membrane that you saw earlier on that structure, we can look for the information that we can gather from these. Like I said, it can be from the surface. So on the outside, we can use methods like an antibody-based pull down or something called flow cytometry. And that information would be based on what's happening on the outside of these vesicles. So that's one type of information that we can use to look for what we want to look for. We can look base our analysis on something that's contained within the membrane bilayer. And we can use, again, this would be an analysis of the intact exosome, the intact vesicle, the entire structure that we go after. And we can form these type of analysis, including electron microscopy and a particle tracking analysis that will allow us to count them and look at them to see exactly what protein they have on their surface. Finally, we can look at the cargo that's inside them, right? And as I mentioned earlier, there's so much that's available. And so depending on our questions, we can look into their proteins and their lipids and metabolites, and we can go after mutation detection that are specific to the questions that we have. We can do sequencing that are obviously more elaborate, and they give us more information, and etc. So a lot that can be done with this, and indeed very useful for what we're looking to achieve here. So in gliomas, like I said, we are using this platform that is very useful if it allows us to bypass being the tissue biopsy in these patients. And the fluid that we go after here is really plasma, but CSF, you can imagine, a little bit easier, but also a little bit more invasive. And some studies also have used saliva. In our case, it's mostly plasma, just because of the nature that is less obviously invasive than CSF. And we hope that that can be something that we take forward. So in our case, we're using multiple analytes, as I'll show you in a little bit. One of that component, as I mentioned, is these extracellular vesicles, because they carry so much information. And we're using that hopefully to look into this specific molecular characterization. That means exactly what are the features of this tumor, how different is it from another one, and these are the markers that I'll briefly mention to you. So our workflow typically is a patient present with some symptoms. We can send these patients to our study, and they participate in our studies, and then we collect their blood before their surgery, and then we take their vesicles or the cell-free DNA, and then we extract that, and we run something called droplet digital PCR to look for these mutant events that are coming directly from the tumor. So specifically in gliomas, they're very challenging to work with. They have a very poor prognosis and a median survival of only 15 months from the time of diagnosis. And all of this, obviously, it's also a very poor performance. There is a recurrence, there is a lot of treatment, so 15 months of not a very good quality of life, unfortunately. There's poor treatment response due to a very high tumor heterogeneity, and this is true for a lot of tumors, including for gliomas. And the current gold standard, as I mentioned a little bit earlier, is this invasive tissue biopsy. So that's really the only way to confirm that it's truly a tumor. And then in an ideal world, like I said, we would like to move this forward a little bit, where we develop these drugs that are targeted for non-invasive testing. So I'll briefly mention these two publications that we have. One is 2021 and one in 2022, where we work with TERT and EGFRvIII, both highly relevant for gliomas, as I showed you, as I mentioned earlier. A little bit more about TERT and EGFRvIII here. So TERT, this gene is important because it's actually, it's considered mutations in these genes. It's one of the hallmarks of cancer. So it really is a really common mutation that happens in a lot of cancer. And this is really how the cancer cell survives, by being able to change the way that this gene works, and by elongating that structure that normally it ends after a while, after 20 cycles or so, and it goes into senescence, and that's when we all get old. But in cancer cells, they're able to change the pattern here, and this TERT region is able to amplify constantly. So that tumor cell really never gets old. So this is one of the hallmarks of cancer. And so it's a common mutation that happens in many cancers, and including gliomas. And in gliomas, it's in high-grade gliomas, it's actually as high as 80%. And there are two typical changes that we call mutations here that occur in this gene. And the second one is this EGFRvIII. This one occurs at the gene level, so at the DNA level. This EGFRvIII, obviously the changes at the DNA and RNA level, but the endpoint here is a protein that is changed. And so if this is the lipid bilayer of a cell, this is how a normal receptor would look like. And in this case, this EGFRvIII is deleted and you have a shortened version, which biologically just translates into a receptor always being turned on, we call. And so it's always responding to stimuli from the environment. And that means that the cell is always thinking that it has to grow. And that's how you end up with a cancer in this case. So I'll just briefly tell you about what we've been doing with these two mutations. This just to give you an idea that, for example, these are grade 2 and grade 3. So although two gliomas are pretty devastating, we can still try to improve their outcomes by diagnosing them as much as we can, or by understanding the tumor as much as we can. So, for example, if we're looking for if a patient is only TERT mutant, we know that this is survival curve, survival percentage. So in years you see below here. So if a patient is only TERT positive, it has the worst survival. But if a patient is TERT and IDH1 positive, then it has this better overall survival that you can see here based on these curves. And while that's less true in grade 4, for example, and so these subclassifications, subtyping of these type of gliomas would be so relevant and so important if we were able to do this in blood. So some of the things, as I said, some of these mutations, obviously they're very interesting to look for, but not very easy to develop these assays that can work for them. And one of the things that we found actually with both of these mutations is something called G-enriched area. So this is an example of one of these structures, and both of them are very similar. So I'm going to highlight some of these aspects. So you see G, 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 and G's and G's, lots of G's and C's, and even more if you look at the GC repeats. And so what we know about these GC repeats is that this is something that it really messes with PCR amplification. It messes with RT. It messes with all those tools that we have available in our toolbox to look for these structures. And so far, we had no way of looking for these sequences using what we call standard PCR. And these GC structures, by the way, also affect sequencing and really all the downstream applications that people have been trying to use for these genes. So, and this is another way to visually look at how these bonds happen. So those secondary structures means that different base pairs, like AU, GC, as you hear, they like each other too much and they end up fusing together. And ideally, we would like that structure to remain linear and open up so that enzymes can go in and read it during a PCR. But instead, you have all of these pairing that happens that you wouldn't want to happen. And you can see visually here a difference between something called EGFRvIII, which in this case is a mutation, and something called EGFR wild type, which is the normal sequence. So obviously a little bit of it happens normally, but you don't want too much of that base pairing happening. So in this case, we ended up, after looking for many ways to improve this amplification, we ended up with this additive called 7DG, which is in this case a modified nucleotide G. So it's a G, but it's slightly modified, that will incorporate into this new product that we're generating during a PCR. And when you do that, these structures remain stable and therefore are able to be amplified and to be detected. And you can see here visually a difference between a PCR for EGFRvIII in this case. If you add 7DG, you see all these dots, which mean mutation. But if you don't add 7DG, you have a drastic drop in detection of this sequence. So 7DG turned out to be very helpful for the detection of both of these mutations. And since both of them are upwards of 85% of G enriched regions, G and GC regions. We also did other optimizations, obviously. We looked for temperature and other additives to make sure that we have a good and robust assay, which is something that you want to generate in case of these patient samples. So that once it's ready for clinical application, it's robust and it's something that we can trust, obviously. So what did we do now with these assays? So an example here is looking at EGFRvIII in tissue. So this is cohort 1, and then I'll show an example of a cohort 2. So in this case, we had 20 tissue samples. And so this is not even plasma yet. This is just looking at tissue. So our question was, does this improved assay with this 7DG, does it help us detect a higher rate of EGFRvIII even in tissue? And the answer is absolutely. So you see here, when you add 7DG versus non-7DG, we detect 17 out of 20 of these tissue samples that have the EGFRvIII mutation. And if you don't add 7DG, only 7 out of 20 turn out to be positive. And 7 out of 20 is about 24%. In the literature so far, this mutation has been reported at about 30%, 35%, if you have a cohort of patients that are EGFR amplified. So if you have an EGFR amplification, that means you have a lot of this EGFR. You have about 40% chance that you will have this EGFRvIII deletion. But in this case, in our case, it seems to be a little higher with an optimized assay. And similarly, we looked at the second cohort, and we did the same thing. In this case, we had 17 patients, and we add 7DG or we omit 7DG, and you see that when we add it, 13 out of 17 turn out to be positive, while only 6 out of 7 are positive without 7DG. Again, showing that we think we have a much higher prevalence of EGFRvIII for these patients, which is relevant for so many reasons, as I said, for subtyping this tumor, for better understanding, for improved clinical care, but more importantly for treatment options, right? If there is an EGFRvIII drug out there, which there are, especially because, as I mentioned to you, this is on a surface, right? So it's on the outside of the cell. And so a lot of therapies are being developed. Some of them you may have heard called CAR T cells that can target these tumor cells. So if we know that, 80% of patients have this deletion as compared to 15%, obviously we would have a much higher population of patients that can benefit from the treatment, but also can help to understand that treatment itself. So finally, we went to plasma, which is our main goal. And so how did these two assays perform in plasma? So first, I'm just going to show TERT as a summary table here, because that's the first one we published a couple of years ago. So in this case, we ran 157 samples, and we had a total sensitivity of 77%, specificity of 90%. And you can see that shown here with a pretty high negative and positive predicted value. And again, this is 80% of patients that have it. So, and an 80% sensitivity in plasma is very exciting for this type of, for this disease. For EGFRvIII, similarly, and TERT, by the way, we took the same approach, several cohorts. In this case, 2 and 3 were blinded to the operator, and our controls include patients that that are EGFR amplified also, and we also include 14 healthy, and 30 of them have the mutation based on tumor tissue. And so for EGFRvIII, we end up with a 73% sensitivity and 98% specificity, which again, it's very, very exciting for these type of mutations, for these challenging mutations and also for this challenging disease. We also show that we can monitor these mutations in longitudinally, and this is similar also for TERT, but here I'm showing EGFRvIII. So if you have a patient that ends up with a treatment response, that means that these are months post-operative. So GTR here means gross total resection. Patient comes in, the tumor is removed. We get that blood sample right before the tumor is removed, and we're able to detect that mutation here. This is showing copies in plasma. After that surgery, patient undergoes treatment, temozolomide radiation, and then 4 months later, the patient comes back. MRI shows stable disease, and indeed our assay also drops below detection. Similarly, if there's a recurrence, gross total resection, that patient undergoes same treatment. Unfortunately, that patient does not respond. There's a second dose of temozolomide treatment here that happens for this patient. Patient goes into surgery again for gross total resection. We receive that sample and we confirm that mutation is still there, unfortunately. So how are we taking this into the future? I'm showing you 2 mutations, obviously. We would like to expand this to 3 or 4 different mutations that would allow us to completely subtype these tumors. And in this case, we're using something called QIAcuity, and it's a neat system because it allows us now to combine 5 different mutations. And we're very excited about that. And that's what we are spending a lot of our energy on right now. And this is visually just to show you that we can look for the target, digital PCR also in this case, which allows you to individually detect these events into individual droplets. And you get this absolute quantification, again, that tells you you have 2 copies, 5 copies, 10 copies. These are very, very sensitive ways to look at very rare events. And so this is a little bit of an overview of what we would like to do. So the goal would be rapid turnaround to inform that clinical decision-making, use them as companion diagnostics, and also cost-effective because you're really only looking for 3, 4, 5 mutations by PCR, and that that would be targeted understanding of the disease. And so again, if we're able to detect that tumor, isolate, when there's a tumor, we isolate, we run the PCR, and within an hour or 2, we're able to tell that patient that there is or there's no signal for that specific mutations, similar to what's being done today for regular blood work. And this is a little bit of what it looks like when we combine these mutations. And right now we try to say, if I put, a higher amount of this mutation and a lower of this one, and then I change that amount from 1.2 to 2.5 to 5, how does my signal change? When do I lose signal? When can I rely on it? And those are the questions that we're really trying to understand. And finally, as I mentioned, if we can develop these multiple analyte assays for the simultaneous blood-based detection of EGFRvIII, TERT, and IDH1, as well as a couple more that are relevant in this case, we would be very, very happy. And obviously that's where we're hoping to get one day. And just to conclude, so we are able to do this minimally invasive diagnostics to aid their clinical decision-making and disease monitoring, surveillance, and importantly, as I mentioned, to aid the development of targeted therapies such as CAR T cells that would otherwise not be possible if any treatment asks you to go back to the patient, to the tissue every month to be able to get that biopsy to confirm if the treatment is working or not, or if that mutation that you're targeting is still present or not. As we know, these tumors evolve quite a bit. It wouldn't be feasible, but with a liquid biopsy, you can do that frequent monitoring and determine if that patient would still benefit from that treatment, and et cetera. And finally, I want to thank our funding sources, the Rappaport Foundation for supporting us for this work, and Mahin Batul, who worked on the EGFRvIII assay, and Kaushik, who worked on the TERT assay. And thank you for your attention.
Thank you so much, Dr. Balaj. We do have one question that just came in from the audience. Do normal cells also release information in the blood?
Yes, so that's a very good question. So obviously here I'm talking about cancer cells releasing all of this information. but you can get information from normal cells as well. And sometimes, usually, that's actually a problem because it can hinder your signal, right, from your tumor cells. And sometimes you're lucky enough that that signal, in the case of EGFRvIII or TERT, is different enough that it wouldn't happen in normal cells. So you're looking for something that, if it's there, it's clearly a tumor response. But if it's not there, then you know that there shouldn't be a tumor, right? But if sometimes these mutations, or we call them gene signatures, they're not exactly, they're healthy people have them as well. So it requires a lot of patience, a lot of replicates, a lot of work to be able to say, this is specifically to a disease, and this is something that happens in normal patients, normal people as well. And so, yes, this information is found. Some of it obviously is less, it shouldn't be like some of the, cell-free DNA, some of it should be less pronounced in a healthy individual, but they are secreted, as we all have cells that continually grow and divide and die even in a healthy person. So.
All right, terrific. It looks like that is all we have for questions for you at the moment. Thank you very much. I really appreciate your time today. That was a wonderful presentation.
Thank you.
I am going to move our presentation to Dr. Mukherjee. Doctor, we should be able to see your screen now. Okay, wonderful. I think we can see your screen.
Great. I just can't start my video, Sean. Oh, there we go.
There you are. Okay, terrific.
Wonderful.
I'm going to go off video and we'll start your video.
Fantastic.
Okay.
So thank you, everyone, for joining us today. I really want to thank you, Sean, for all your help in crafting our session, and then to Phyllis and Jerome Lyle Rappaport for their dedication to junior investigators. I'm delighted to share some of our exciting preliminary findings at the very end of our talk that really was not possible without the support of the foundation. And so I've been interested in neuroscience for about 20 years, and for the last decade, been really truly fascinated around questions about how we as humans respond to an infection, but really why our nervous system has to be so integrally evolved in some of our emotional responses to infection. So I suspect that some of you in the audience can remember a time when you've been infected and you felt tired, fatigued, and perhaps a little depressed, and maybe that's an protective or an evolutionary mechanism to heal. Well, these infections actually trigger a very intense emotion, like extreme depressive symptoms or extreme anxiety or cognitive dysfunction or blunting. And so for a portion of those individuals, their neurological function actually does not return to a baseline after the infection's over. They don't return to homeostasis, even if the infection does not involve the brain itself. So my name is Shabani Mukherjee. I'm a neurologist. I really am trained in caring for people with infectious diseases and neurological complications. And so my lab is centered around sort of the suite of some of our adaptive and maladaptive neurological responses to chronic infection. And the fact that the brain is somehow integral and part of the immune response to an acute infection is not novel. We've known this for centuries. And we've known that there are a variety of adaptive behaviors that occur in response to an inflammatory cytokine or a chemokine to an infection. And these responses, behavioral responses, are actually conserved across species. So if you take, for example, mice, when they are challenged with some sort of immune challenge, you can identify that these animals will actually spend and have changes in their sleep or changes in the phases of their sleep they'll spend more time in non-REM sleep. And they really do so at the expense of more restorative REM sleep behavior. And so this results in a change of wakefulness. And we can somehow assume that this is beneficial. And indeed, there's actually a robust change that occurs with some of the neuroendocrine responses to an infection that can then also affect how our peripheral immune system responds. And we term this types of behavior as part of the acute sickness behavior. So this challenge that we are faced with is that in some certain cases, we have individuals that have chronic inflammation or chronic immune activation. And these behaviors that we think are beneficial somehow become maladaptive, and they last a lot longer. And my lab really particularly focuses on people with HIV. And we've made extraordinary, incredible strides in treatment of HIV by creating very effective antiretroviral therapy. But one of the challenges is that individuals still have to chronically live with the virus. It's not curative. And so even that small amount of virus, even if it's not replicating, can trigger a host of immune activation responses in certain individuals. And that could potentially lead to some individuals having neurological disturbances. And we've shown this for sleep disturbance. We're showing this for pain, as well as cognitive blunting or cognitive fogs. And I'm going to talk a lot about how some of the persistence of immune activation can relate to depression and potentially some neuroendocrine changes. The one thing that I do want to point out in sort of the schematic is that while I've made this sort of simplistic, we've got to understand that in the human condition, things can be, in fact, complex. And so part of the discussion when we're talking about a little bit is how we think about this in humans and not so much animal models, where there can be some things that are bidirectional. And I'll just put this case in point. When we think about animal models of stress, for example, and looking at mice, we can have individuals that have depression actually leading to behaviors that look sort of stress models, excuse me, that lead to depression-like behaviors, depression or sleep disturbances. And then you'll suddenly see sort of these elevations in cytokines. So inflammatory cytokines that we think traditionally, sort of something like TNF-alpha or IL-6, or even chemokines that act as if we want the immune system to respond, things like monocyte chemoattractive protein. And so even in scenarios where there's no infection, just having the depression can then somehow lead to elevations of an inflammatory response. And so in that setting, I want to think about, when we think about people with HIV as well as people with depressed mood, we want to understand if in part of their depression, is there an inflammatory subtype of individuals that may benefit from some therapy? And so there is data now that's out there that's correlational in large part to sort of think about, yes, individuals, when we look at blood, and you take a look at some of the cytokines, I'm just showing one, IL-6, you can see some relationship with depressive symptoms, just shown on the severity score. But one of the challenges, again, with working with individuals is that not everybody who has HIV is going to be depressed. Not everybody with HIV with inflammation are going to have depression. And you can sort of see this here. Again, these are all individuals with HIV virally suppressed who have depressive symptoms. And some of those individuals do, in fact, have high IL-6 levels. You'll have a certain set of populations that don't have elevated inflammatory cytokines, but still have depression. And then you'll have a subset of individuals that have elevations of their inflammatory cytokines, and they don't seem to be depressed. And you can even divide it further into how heterogeneous some of these discussions can be. When you look at and divide it out by something like sex, some of the inflammatory signatures that you can see are not the same as what you'll see in women as you see in men. And the relationship of how inflammation and how cognition are affected can be different into these two groups. And so a couple of challenges that we face in our lab and as in our field is who are the people who may have an inflammatory type of syndrome, who have HIV and depression, so an inflammatory biological subtype of depression. And if we can identify those individuals, what are the treatments? Because it may not be so easy to just give an anti-inflammatory. I'm going to answer the second challenge first. Do we have something in the arsenal for individuals that do have an inflammatory biological subtype of depression, if we do believe there is one? And the answer is really not. So many people with depression really do struggle because most of our first line antidepressants really are targeting one type of system and sort of the monoamine. So we've thought about serotonin, we can think about norepinephrine, but there's really targeting monoamine metabolism, and that's usually first line. But about a third of individuals, irrespective of whether or not they have HIV, don't achieve symptomatic control of depression with some of the first line therapy. So we really do need more out there. And there's really nothing out there that specifically targets immune activation, even though the concepts of immune activation as a trigger for depression has been out there for a very long time. And so we are not able to target things like inflammation within the central nervous system that does influence how much of these neurotransmitters are available and released. It doesn't target other neurotransmitters that are really disrupted in people with depression or anxiety. And nor does it really target any of our current first line on some of the inflammation that can occur in depression, and then that downstream struggles that we have with neuroplasticity as it relates to inflammation. So there's much to do. The question then for the first is, who would benefit from an antidepressant that may not be first line, but may be targeting either the immune system or other neurotransmitters? And so that's where the bulk of my lab has worked on, is trying to find biological subtypes. One of the biological subtypes that we had looked at very early on in people with HIV was with my mentor who was at the Dana-Farber. And so one of the platforms that we had used was really thinking about using untargeted metabolite profiling. I'll talk a little bit more about this, but essentially what we're trying to do is find targets in people with depression who have HIV, and to try to see if there are other biological mechanisms that we can use. And in this study, what we looked at was individuals who have depression, at least at one visit, who had HIV, and it looked at and compared it to people without depression at a visit who also have HIV and don't have depressive symptoms, and looked at the plasma marker profile of those individuals. And we did this for several cohorts. And what we in bulk found was that individuals with HIV and depression really do have alterations, not only in tryptophan metabolism, which is a bulk part of the monoamine pathway, but they also have decreases in cellular energy or mitochondrial energetics. And what Dr. Gabusta had shown previously is that we do have some individuals that do have elevations in inflammation that includes elevations in CXCL10, which you can see in the blood, other elevations of other inflammatory chemokines like CXCL11, as well as metabolites that we know are associated with either immune activation or death in people with HIV. And so with this, we were interested in trying to understand whether or not this biological subtype not only is for people who are acutely depressed, but whether or not it really fits for those individuals who are persistently depressed, who we may want to target with some sort of therapy. And so we used a similar type of profile where we definitely looked at individuals who had persistent depression, looking at plasma profiling in people with HIV who are virally suppressed. And what we use is a platform called untargeted metabolomics. And what that really involves is taking blood from individuals and looking at mass spectroscopy of different types and trying to find metabolites that are produced in the blood. And one of the reasons why we like looking at the metabolome as versus other types of omic type of platforms is that it's probably the closest to the phenotype that we're interested in. It is also influenced a little bit by diet and other microbes. And so understanding the metabolome can help us understand potentially some biological targets that are closest to what the phenotype that we're interested in. And so in our case, in this older study, we looked at those individuals who had HIV, who had persistent depressive symptoms. And what we looked at is really people who really met threshold for really high depression, really above the clinical threshold that we would think about for depression. And they had to have done so over time. So they had to have a longitudinally depressive symptoms. And we tried to compare it to their extreme. Those individuals who had HIV are virally suppressed, but really did have very minimal symptoms. And what we looked at is trying to find some sort of targets. We had two different cohorts. One cohort that we looked at, all of which had, again, HIV and age-matched individuals with and without depression. We could identify different types of metabolites that were different between these two groups. We looked again and used a separate cohort, which we call the replication cohort, and again, see the differences between groups. Fundamentally, what we're looking for is patterns between these two cohorts. And ultimately, what we ended up identifying is in people with depression who have HIV, there's these decreases in cellular energy, these acylcarnitines, the same type of cellular energy that we saw identified previously. So if you do have depression with HIV, there's clearly a decrease in some of the energy metabolites that we have. But what was a surprise to us, and I think of extraordinary interest, is that we also saw decreases in some of our neuroendocrine mediators that we can identify in blood. Some of those, things like pregnenolone, some things like DHEA, were decreased in those individuals who had chronic depressive symptoms. We also saw decreases in some of their downstream sex steroids as well. When we look at our replication cohort, we can see not only are there decreases in some of these similar sort of cellular energy metabolites as well as these neuroendocrine mediators, but they do so in sort of the exact same pattern, where individuals with chronic depressive symptoms have decreases both in cellular energy and some of these neuroendocrine mediators called neuroactive steroids. We went further to try to take a look and see whether or not we can identify some type of biomarker that we could reasonably use in a further trial to take a look at people with HIV who may have a biological subtype that might be amenable to treatment. And so we took a look at some of the other hormones that we know we can assay in blood, the most common being cortisol, which is a stress hormone that's been shown to be increased in people who have depression. In general, and the basic gestalt is that these individuals don't have elevations in cortisol. What they do have is decreases in DHEA. And what you can use, and people have used, are types of ratios where you can think about seeing whether or not individuals with depression could have abnormal levels of this type of response with either high or normal cortisol or low or normal DHEA. And in our cohorts, we could say that we could identify those individuals with depression who have HIV using this biomarker. But it's not enough. It's not actually specific enough. If you add some more of the neuroactive steroids that you find in the blood, you can get a little bit better with your specificity. But in truth, it's not enough to start providing a treatment for individuals. We really do have to understand the pathway a little bit more to identify biomarkers. And so one of the things that we're very interested in trying to understand is whether or not we can use this neuroactive steroid pathway as part of understanding not only the biological reasons for why this group of cohort of individuals may be depressed, but whether or not we can use this as a treatment option. And so what are neuroactive steroids? So neuroactive steroids are cholesterol-derived hormones. They're really capable of binding neurotransmitter receptors, and that's why they're called neuroactive steroids. They're fundamentally produced in the adrenal glands, but we see them locally produced actually in the brain, where they're called usually traditionally neurosteroids. Fundamentally part of the stress pathway, and they're downstream a very common pathway from the brain to the pituitary to the adrenal axis, so the HPA axis. What we know and understand about their function is that they do affect mood. So neuroactive steroids are known to both affect mood, they can affect cognition, they can affect different neurotransmitters than the ones that we think classically that we traditionally target for depression, including serotonin. So they target things like glutamate as well as GABA. They don't traditionally bind to glucocorticoid receptors, but they can affect immune responses, which makes them somewhat interesting for us as we think about immune subtypes of depression. One of the key things that is interesting is that some companies there are clinical trials looking at whether or not if we supplement or give neuroactive steroids, do you get some improvement? And I'll talk a little bit about some of the newer FDA approved therapies that use neuroactive steroids as something to use for clinical disease. But one of the things that we had realized as we were trying to understand neuroactive steroids is that we have very limited understanding about what is going on in people with HIV just at baseline. And one of the reasons why we have some difficulty in understanding what neuroactive steroids are doing in a population like people with HIV is that it's very actually challenging to resolve many of the neuroactive steroids from each other because chemically they look very similar. And the key thing is their functional effects are dependent though on their structure. So if you take the parent neuroactive steroid pregnenolone, what you can see is that it itself may not have actually activity on some of the neuroactive steroid neurotransmitters, but its downstream metabolite does. And the differences are really based on some of its steroids changes at the steroid rings. If you look downstream from pregnenolone, downstream to the pregnane neuroactive steroids, those neuroactive steroids tend to have very specific effects on GABA A, and in large part, they have different structures than what you see from the parent neurosteroid. So it's extraordinarily important to try to resolve and understand some of these neuroactive steroids. The reason why I think it's really important that we do so, especially in people with HIV, is twofold. One, we really have recognized since the 1940s that some of these neuroactive steroids do have some benefit for mood-related disorders. So if you take a look at pregnenolone, these have shown some safety and benefit for healthy control individuals for both mood and fatigue. Downstream, they've had some benefit for allopregnanolone especially to look at pregnancy. And in fact, the FDA just approved an IV formulation of an allopregnanolone type of molecule for postpartum depression. There are several trials looking at the parent neuroactive steroid for other disorders that we're interested in, including in pain as well as depression in cohorts that are not HIV. As I had mentioned, we're very interested in trying to understand whether or not we can identify something that affects maybe neuroendocrine type mediators, but also affects the immune system as well. And what we've known that for a very long time, since the 1950s, is that neuroactive steroids, including pregnenolone, has been used in inflammatory conditions that have immune activation chronically. So that includes rheumatoid arthritis as well as lupus. And in part, are not used because they're not very strong. There are stronger drugs that do suppress the immune system. But what we have been learning more and more recently, especially as the FDA is starting to look at this drug class, is that both pregnenolone as well as its parent downstream metabolite allopregnanolone can affect some of the cells that we're very interested in understanding that do contribute to immune activation in people with HIV, as well as influence some of the CNS or central nervous system inflammation cells called microglia. And so for that reason, we're very interested in trying to understand neuroactive steroids. And it's with the help actually of the Rappaport Fellowship that we were able to collaborate with an individual who is very well known in the neuroactive steroid field. And not only is she known for neuroactive steroid quantification, she does so in human populations and in blood, where it's really hard to resolve some of the structural differences between these metabolites, and she uses mass spectroscopy. And so with the Rappaport Fellowship, we were able to look at several of our clinical cohorts that we already have developed in people with HIV and compare it to clinical cohorts that we have that are age matched with people without HIV to take a look at what are some of the neuroactive steroids in blood at baseline, irrespective of depression. Our hypothesis prior to doing this was that we would have anticipated people with HIV who are virally suppressed, relatively healthy, would not necessarily have differences between neuroactive steroids, but that was actually not the case. So all the steroids that we were able to quantify are shown in pink. And what we can see is that the parent neuroactive steroid is similar between groups. The DHEA that I'd shown you had differences, also similar between groups, as well as the sex steroid. But what was not similar, even at baseline, irrespective of depression, is some of the pregnane hormones. That includes pregnenolone as well as allopregnanolone, which is decreased in people with HIV. That why we are decreased still remains unknown and something that we're extraordinarily interested in trying to identify. As I had also mentioned, that many studies had shown that there is some relationship between neuroactive steroids as well as inflammation. And for us, we were interested in understanding what that relationship could be in people with HIV. So if we looked at our total cohort that we had already assayed for neuroactive steroids, but we had availability of inflammatory cytokines, we can see that there's an inverse relationship between neuroactive steroids as well as inflammation. So what that means is for pregnenolone, for example, if you have low levels of this neuroactive steroid pregnenolone, you may in fact have higher levels of inflammatory cytokines. That includes higher levels of IL-6. You may also have some higher levels of TNF-alpha and some chemokines that we're particularly interested in that affect monocyte migration, including MCP1, as well as soluble CD14. This also holds true for allopregnanolone, that chemokine, that neuroactive steroid that's lower in people with HIV. There is that same inverse relationship, predominantly with markers of MCP1 as well as soluble CD14. We can show that in people with HIV, just looking at that group, we can see those similar relationships with pregnenolone as well as allopregnanolone. So our group is actually extraordinarily interested in trying to understand whether or not this pathway could not only help affect our inflammatory questions, but whether or not that also affects behavior. And again, with the Rappaport Fellowship, what we were able to do is take a look at a second cohort. That cohort is of people with HIV, but those individuals had brain MRIs as well as mass spectroscopy, so where we could look at neurotransmitters. And so for our early work, we were able to show that allopregnanolone has this relationship with soluble CD14. Now that's in the periphery. We had previously shown that that soluble CD14 that we can see in the blood has a relationship, a direct relationship with excitatory neurotransmission. So there is some increases in excitatory neurotransmission in the insular cortex of people with HIV that's related to their soluble CD14 levels. What was interesting to us is in that same location of the insular cortex, we can see that the inhibitory neurotransmission GABA is associated with depressive symptoms. And so this triangulation is what we're interested in to try to understand. Right now, we don't have causation that allopregnanolone or pregnenolone has any influence on any of these neurotransmitters or in behavior in people with HIV. And so what our goal is to now future look at is to try to understand whether or not neuroactive steroids do affect our neurotransmission, does affect depressive symptoms, and what is the effect on immune composition and function, specifically in people with HIV. And so what we were able to then achieve is to start a phase 2 randomized double-blind placebo-controlled clinical trial. We're specifically focusing on people with HIV. We are focusing on those individuals that do have depression, and we're taking a look at the parent neuroactive steroid called pregnenolone and trying to understand the relationship on GABA before and after pregnenolone. What's the relationship on behavior, sort of the incentivized reward, as well as depression? And also taking a look and seeing if there are changes with immune composition and function. And we're going to do that using flow cytometry, really focusing on monocytes, the innate immune system, as well as looking at many of the inflammatory cytokines, as well as transcripts that occur with that can happen with monocytes using RNA-seq. And so I'd hope at some point in time we could talk to you a little bit more about some of our findings after this clinical trial. But with that, I want to thank everybody in my lab that has really done a lot of work. Those are individuals, currently Emily Rudman, as well as Rami Kashan, as well as those individuals who are part of our clinical trial called Soothe. I want to particularly highlight Christine Marks, as well as Janet Lowe, who had helped with the clinical trial that we used some of those samples from, and my own mentor, Donna Gabuzda, as well as the Rappaport Foundation for all their help and support. And most importantly, we want to thank all the patients and caregivers who contribute to our clinical studies. Thank you for your time.
Thank you, Dr. Mukherjee. We do have a question here that I will share. Do infections like influenza and COVID influence behavior and mood? And can we find what we find with HIV and other infections?
It's a great question. Thanks, Sean. I was going to say that, you know, the devil's going to be in the details. So things like HIV, HIV is a unique virus that integrates into the DNA of its host. And so that's one of the challenges and one of the reasons why you see chronic immune activation. And in infections, RNA infections like influenza or even COVID, what you have is you don't have known integration at this time into the genome. But one of the challenges is that you can get with influenza and as well as COVID reinfection. So you might have repeated acute behaviors that may over time develop into some maladaptive responses. We don't actually know that yet. So I think that devil is in that detail. What we hope, though, to understand is whether or not what we identify in people with HIV, we can see, and whether or not we identify treatments, whether or not that would then work for people who have other infections and have some of these chronic type of behaviors or what we think would be maladaptive behaviors. And so I think what we learn from HIV could be then needed to be studied in those populations of individuals. You asked a second question, Sean. Can you repeat that for me?
Can what we find with HIV work with other infections?
Yeah, so I think that sort of addresses, I think, a little bit, our hope is that what we identify with HIV does work in other infections. Not only do we find some sort of biological signature, we can then try to use that for looking at different cohort populations.
Terrific. Well, I want to thank you so much for your presentation today. It was very enlightening. And thank you both. We are so proud to have you as Rappaport research scholars. And thank you all for joining us here today. And we hope to continue this series moving ahead in the future. Thank you very much. And thank you for joining us.
Thank you.
Thank you all.
Thank you.